Well, good morning. It's truly a pleasure to be here today. And as Steve mentioned, I like to do experiments. I'm a scientist. And you might expect, as a professor, I teach classes. And one of my favorite things to do is get the class involved in asking questions and having discussion about topics that are of interest to them. So I hope I don't make you uncomfortable today. But what I want to do, which is my assignment, my primary assignment, I'm a resource for all of you. A little bit more of my background, and Steve gave you some of the details. I've been doing research at the University of Minnesota on DDGS for about the last 15 years. And it's a fascinating ingredient. It's not only just a package of nutrients that has value in all species of food animals that we feed, but it also appears to have some interesting and unique properties. And we're going to talk about some of those things today. But again, I don't want to make it a lecture. I will if I have to, but I really encourage you as we go through the day. And by the way, I'm going to be here till Sunday morning, so we've got 2 days to talk about DDGS if you'd like. But what I'd really encourage you to do, because I know you all come with different experiences, you have maybe many questions about concerns or issues related to DDGS, I want this to be an interactive question-answer session primarily. And if you're comfortable with that, I'd be really happy about that. If we run out of questions, we're going to go back and talk about some of the prepared slides that I have. So we're going to do a little bit of a different format, a little different experiment. I was thinking last night as I was visiting with some of the folks, some of you at dinner, I often like to start off by reminding ourselves that every one of us sitting in this room, me as a researcher, you as ingredient traders, maybe nutritionists, people that are supplying feed to the animal industry, we're all involved in the food production business. And as Eric mentioned, I understand and appreciate very much, the aflatoxin scare that occurred here in Europe last year. And that is a concern because those of us involved in the food production business are concerned about food safety and feed safety, which is directly tied together. And so there's no wonder that I think, at least what I've been hearing the last 2 days, one of the biggest concerns that many of you have is, what is the risk of aflatoxins or mycotoxins in general in DDGS? All of you are involved in business, and you all understand and try to manage business risk. And that's a noble and very important thing to do. And so mycotoxins, you're right, is a risk factor, but I want to spend a little time talking about mycotoxins and what we know about them and how we can manage these risk factors. So I just want to start off by reminding us that food security, food safety, and feed safety are all interconnected. Another interesting statistic, and these are just general things before I start getting into the details, and hopefully we'll start asking some questions. Feed safety does have a significant impact on our global food safety system. And I don't know how many of you might have realized this, but there's somewhere over 100 kilograms of feed consumed by animals, and the animals, of course, or animal products, are consumed by people a year. So feed plays a very big role in our global food safety system. And certainly, we all understand, many of you have experienced this in your own businesses, if you're involved in the feed business at all, that if there is a contamination issue, let's use aflatoxin as an example, it affects not only your business, but the entire food chain. And so we're always interested in trying to minimize these risks because it costs a lot of money in that entire food chain. So to get us started, what I decided to do, instead of ending the discussion today talking about mycotoxins, I wanted to start with the discussion about mycotoxins. And I'm doing this primarily because Number one, DDGS is no different than any other feed ingredient. It's a package of nutrients from a nutritional perspective. Protein, fat, fiber, minerals are all in different proportions relative to other ingredients, but as nutritionists, we understand that, and our job is to put a combination of these ingredients together in the right proportions to meet the animal needs that we're trying to feed. It's really that simple. I mean, it's more complicated than that, but fundamentally that's what we're trying to do. And as we do this, one of the things that we're trying to avoid, or at least recognize, are what are the risk factors of any ingredient that I'm using. And so I thought it'd be good to start our conversation this morning talking about what I consider to be the 4 major risk factors associated with DDGS for various species. The first one, of course, is mycotoxins, and we'll talk about that a little bit. The second one is that some sources of DDGS have high sulfur content. Why is that a concern? Well, if we feed, like we do in the US, beef cattle, for example, if we feed those beef cattle 40% of their dry matter intake is DDGS, and if that DDGS source has over 1% sulfur, and if the water those cattle consume has high sulfate, and the forages they eat are also high in sulfur, we run the risk, depending on the total diet concentration, that's water, forage, and the grain, we run the risk of causing what's called polioencephalomalacia. It's a kind of nervous disorder that kills cattle. So it's a sulfur toxicity, let's think of it, for those of you that haven't had experience with that. So that's a concern. Is it a reason not to feed distillers' grains? Absolutely not. With all these risk factors, there are management strategies to deal with that risk if it exists. I will tell you that of all the survey work that we've done of US ethanol plants and DDGS sources, sulfur content generally is not an issue, particularly if you're feeding low inclusion rates. But sometimes it is, so it's something to be aware of. High sodium. What species do you think sodium content of DDGS would be a concern? Chickens, absolutely. George, you're a very good student. Chickens. Why is that a concern in chickens? If you have high sodium content and you don't make adjustments in your supplemental salt in your diet, whether it's broilers, layers, ducks, geese, whatever, you run the risk of wet litter. So knowing that is important. The last one, corn oil, which is found in DDGS, and of course the trend in the US is to take corn oil out. Much of that's going into biodiesel production. Some of it's going into animal feed, primarily poultry. But corn oil is also high in energy. But one of the characteristics of corn oil is that it's high in unsaturated fatty acids. So if I feed high amounts of DDGS to some species of animal, what species do you think that would be of concern? Some of you that have been feeding DDGS probably know this. Poultry, not so much. But thank you for the answer. Primarily in pigs. Primarily in pigs. Good answer. Why is that a concern in pigs? Bacon. Yes, very good. Pigs become what they eat, so to speak. So if I feed high amounts of any vegetable oil, including corn oil, that's right, it causes the pork fat to become soft, more oily. It becomes more like what they eat. And so if you love bacon, which I do, by the way, the characteristics of the bacon aren't quite as desirable as maybe what we would like as consumers. It becomes softer, stretches more. Shelf life stability might be a concern because food products that are high in unsaturated fatty acids unless preservatives are added, may not stay fresh or may become rancid in a shorter period of time. So in the case of the US, where we feed 30, 40% DDGS to growing finishing pigs, corn oil content is a big concern. In fact, we have different management strategies in terms of diet formulation to deal with that, to meet whatever the pork processors' guidelines or specifications are without negative consequences. So I thought it would be good to start this morning with identifying, if you're going to use DDGS, and I don't care what country you're in, you need to know what the risk factors are. And these are the four that I could come up with. But I want to spend some time talking about mycotoxins to start off, and make a couple general comments, and then I'm going to sit down and we're going to have a conversation. Most of you know that mycotoxins are produced by molds. Molds are present in the environment everywhere. And depending on the environmental conditions that exist, whether it's in a feed bin or in a feed bunk or a feeder for an animal, or in the field before harvest, different molds under the right environmental conditions will produce mycotoxins. Now let me remind you, and this is often a confusing point for many people, just because we have mold present in corn, for example, does not necessarily mean that mycotoxins are present. They could be, we don't know. And just because we don't have mold present in evaluating corn or a feed ingredient doesn't mean that mycotoxins aren't present. So they're related, but they're not in some ways. The second point that I want to remind people, and I see these laboratory tests quite often. People say, oh, I'm concerned about mycotoxins in the corn that I'm buying or feeding to animals. So I send it to the laboratory and I ask them to give me a mold count. Does that mold count, is that useful information? Absolutely not. It says mold is there, doesn't tell you what kind of mold there is, doesn't tell you whether mycotoxins are present, because that's the disconnect between mold and mycotoxins. So if we're really serious about monitoring our feed ingredients for mycotoxins, we have to actually measure them. And of course, there are tests, laboratory tests and quick tests that are used to do that. Some of you may not realize this, but we often focus on maybe 5 or 6 mycotoxins in feed ingredients. Help me name those. Aflatoxin's one. What? Don, zearalenone, T2, HT2, ochratoxin. Small grains, there's a lot of small grains in Europe, right? Barley, wheat. What's the primary one that we're concerned about there? Ergot. So there's a lot of mycotoxins, but that's a pretty short list. That's what, 5 or 6 that we named? There are approximately over 300 mycotoxins present. Many of these haven't even been identified yet. So it's interesting, here we're focused on the things that we know, but there's a lot of things we don't know and don't know how to measure. We don't know what their impacts are. So I use this sometimes because mycotoxin experts estimate that somewhere around 30,000 different kinds of mycotoxins may be present. Part of the reason I'm bringing this up is that, let's pick aflatoxin, for example, one of our biggest concerns. And why is that a concern for us? Why do we worry about aflatoxin other than the government says we have to regulate it?
It goes to the food.
It goes to the food, that's right. And which species of animals are we most concerned about?
Poultry.
Probably dairy. And why dairy? I'm sorry? Ends up in the milk. These mycotoxins are very resistant to about anything we can do to them. They're resistant to pH changes, any sort of chemical treatment, that doesn't destroy them. They're resistant to heat. So they're a real problem. We can't get rid of them if we have them. And for the most part, unless you have more abilities than I do, we can't control the weather as far as how these grains are produced. So if the conditions are right, they're going to be produced. Part of the reason that aflatoxin, unlike these other mycotoxins that we listed, is such a concern is because it causes cancer. It's carcinogenic. And that's a bad thing, of course. But it also is a real concern in lactating dairy cows in particular, because it's transferred directly into the milk. People drink milk, babies in particular, and we want to protect people. We like healthy, safe food. So aflatoxins are a concern. And it's not just in DDGS, but DDGS is derived from corn. So I already mentioned that it's really difficult, in fact, it's almost impossible to destroy mycotoxins. So once we have them, they're there. And if we have mycotoxins in corn, we can expect that as corn goes through the ethanol production process, those mycotoxins are going to be concentrated by a factor of 3. So you can do simple mathematics. If I have 5 ppb, parts per billion, of aflatoxin in corn, I can expect that in my DDGS, derived from that same corn, I'll have about 15 to 20 parts per billion. Is that a concern? Sure it is. But we also have to remember that DDGS is one ingredient that we put into that animal feed. It doesn't represent the whole diet, it's a piece of the diet. So if we have other grains like wheat or barley, triticale, rapeseed, go on down the list of whatever you might be using, chances are those are not contaminated with aflatoxins, so we have a diet dilution effect. So we can tolerate some mycotoxins simply by managing diet inclusion rates, because it's the total diet that the animal consumes, not the individual ingredient. But yes, mycotoxins are a risk factor in corn, and it's really a function of what's the quality of corn going into the production of DDGS. And if you're in Hungary or the United States or wherever, sometimes we have issues because of the climate or the weather conditions, and sometimes we don't. Historically in the US, if you look at all the long-term data as far as aflatoxin contamination of corn, it happens on average 1 time every 10 years. So the frequency is not high. Yes, it's there.
Is that because of wet weather or is it wet weather?
It's a good question. Is it because of wet weather? Generally speaking, where we do have areas of aflatoxin contamination, it's in the southeastern states of the US, because they typically have high humidity, high temperatures, and if they don't receive enough rainfall, it puts the corn crop in a stressful situation, which makes it vulnerable for mold growth, which then leads to mycotoxins. So it's primarily a weather condition sort of thing. But for the rest of the US, as an example, generally it's not an issue unless the corn crop is stressed. So, one of the things I'd like to discuss briefly, and I've asked Eric to do this, is since we're talking about risk factors and mycotoxins are on everybody's mind, I think it's important for you to know specifically what the quality assurance program is at Pannonia and how they manage or deal with incoming corn, what they're testing for, what their standards are, and how they monitor the outgoing DDGS that goes to your feed mills or animal farms and customers that you serve. So Eric, would you just make a couple brief comments
So our system is a system we designed ourselves. We rely on our U.S. colleagues for a lot of stuff, but on this issue, the U.S. really isn't instructive, because if you're an ethanol plant in the U.S. and you have an aflatoxin issue, you blend things away. And the U.S. government will allow you to do that. So the way that an ethanol plant in the U.S. works, as long as your final product has aflatoxin levels below the legal limits, you're fine. Whereas in Europe, the feed laws are such that if there's 20 parts per billion at any step in the process, whatever has 20 parts per billion or more needs to be taken out of the feed chain, the food and feed chain. So we learned about this on a weekend. Everyone came down to the plant on a Sunday when this hit, and we designed a system. And our first reaction was to take we did have contaminated distillers grains, and we sold that to the energy generating industry. It can be used in biogas plants, and it can be burned in biomass power plants. So our first reaction was, this isn't getting out. We have as you know, we have a brand to protect. It's the only kind of branding we've done. We don't do this for ethanol. But so the system that we have now, when we have a potential seller of corn, Schultz team goes out to wherever this corn is stored and tests it. And that really isn't even a necessary part of our system. But we go out and test more just for coordination purposes. But when that corn comes into, and if it's too high, then we won't do the contract. But of course, what we're shown and what we may get may be different things. So when a truck comes into our facility or Cargill's facility, we physically test every single truck. And we do this with quick testing equipment, and we've gone through several iterations of this, but our quick testing equipment now gives the same results as if we send it out to an independent lab. So, and we double check that fairly constantly. So we're sure that our results are the same results that we get from an independent lab. We of course can't use an independent lab for our trucks because we can't keep the truck there for a couple days. But we do require people to be there for about an extra hour. And if the test results come back too high, then we send the truck away. And if they come back under 20 parts per billion, then we also may send the truck away, because in our contracts we can allow for a truck with high aflatoxins to come in, but most of our supply agreements specify a very low aggregate level of aflatoxins from that supplier. And then we test the corn again when, as you know, some of it comes through Cargill. We store our grain there, it comes back over. We also test it as it comes back over. And finally, we also test the distiller's grains. And we have as Jerry mentioned, we know that there's a direct correlation between the aflatoxin levels coming into the system and what comes out. So we're also measuring our levels against the expected levels. So when we started this, we were turning away a large number of trucks every week, and now we turn away less than 1%. So part of the quality assurance process is signaling to the market that you're very serious about this and that you have a very good system in place so that you can't sneak things through. And today we also, so we test our distillers' grains on a regular basis, and then of course when we sell, we're also testing it under our contracts with a lot of you. So we're testing, I don't even know how many times that is. We test an awful lot of times to make sure, and this is a testing program which we will probably scale down in the next coming months when we're confident that the aflatoxin levels are low enough to warrant that. But we're obviously well into the new corn season, and we haven't scaled back our testing at all. So this is something we remain vigilant about, and we spend an awful lot of money on this testing program. And it's the best money that we could spend, because we made a decision early on to invest in the quality control, and the reaction from you, from the market, is that that is incredibly important to you. And a lot of you have also come through the plant and seen this process in action, and we're dedicated to transparency. Anyone who's interested in seeing how we do it, and hopefully even providing some suggestions to us about how to improve it, we only welcome that. So we, as many of you know, by the end of last summer, the northwestern European market in Europe was closed to Hungarian and Serbian corn. And nevertheless, we were still selling because we think because this quality assurance system was both effective and trusted. And so we consider this to be a great experience for our company, but also how we will approach things later on. There is a next challenge that will come along next year or the year after, and we intend to make only one change to how we did things this past year, and that's now that we have close customers, the very first thing we'll do is call you and ask how you think we should address that situation. So, and we also are training someone to be a full-time quality assurance, quality control manager for Pannonia Gold.
Thank you, Eric. So I think one of the things, as I work with various feed companies and feed ingredient purchasers and marketers, is obviously standards are very important, and they vary depending on the supplier and so on. But one of the things I think Pannonia Gold has is they have a quality assurance program in place to help manage the risk of mycotoxins, and that should be important to you. Doesn't mean that they're not going to be there from time to time, but at least they have a system, and you can argue with the management as to whether or not that system is has high enough standards to meet your needs. But I think what Eric described is a very good quality control system designed to minimize that risk and the concentration of aflatoxin and DDGS, and that's very important.
Could you maybe explain to what extent the authorities got involved in this, and did they rubber stamp your quality assurance program, or is it really totally company driven?
No, they rubber stamped it.
First of all, before you answer that, Eric, did everybody hear the question? No. The question that Robert asked was, what was the reaction of the authorities when this aflatoxin issue hit? Did I get most of the question? Eric, go ahead.
Yeah, so obviously the Hungarian law is no different than the law in the rest of the European Union. The authorities wanted to see a system in place. And the system that we have, the authorities say, is the best in Hungary. And so we're used now by the feed regulators in Hungary as the example of the best possible control system. There are no exact requirements in Hungary for how to manage aflatoxin risks. The only requirement is that a company have a monitoring system in place and also show how it reasonably relates to aflatoxin risk minimization. But that's it.
Did it have any knock-on effect? I mean, in the end, you want to operate in the European market, and it's great that the Hungarian authorities, let's say, approve your quality assurance system, but did they do anything to assign this to Brussels and to say, well, we have this great system now in place and we think it should be recognized in other countries?
No, obviously the GMP Plus system incorporates compliance with national law into the standard, and there's an early warning system for aflatoxin for all feed contamination in Europe. And so over the past year there have been a number of aflatoxin alerts. When this hit us, it coincided with a milk delivery to Italy being rejected at the border. So that's what triggered this within Hungary. But it's also, there were Turkish nuts and Serbian corn. It was a lot of different things last year. But there's No, there's no coordinated system for Europe.
Last year, as a matter of fact, the federal government allowed blending of contaminated corn, which has never been allowed before. And I know that's not the case here in Europe. And so that was one way to manage a high prevalence or a higher than usual prevalence of aflatoxin contamination that we had. Now, of course, all that corn is gone. It's been used or fed or something, and the corn crop this year looks quite good. Yields are good, mycotoxin contamination is very low, but that's basically the level, and it's restricted, of course, for use in dairy cows or total diet. Aflatoxin concentration is 20 ppb, so you want to stay below that. And for interstate shipment, it wasn't to exceed 20 ppb, but last year those rules were relaxed. Whether they stay in place, I don't know. Good question. Other questions? Yes?
Does storage affect the mycotoxin levels of aflatoxin and
Does storage affect mycotoxin levels?
Yeah.
Is your question, Dan? It depends on the mold species. Some of the molds, let me just give you a little bit different visual here to look at. The mold species are shown at the bottom of this slide. Aspergillus, Penicillium, Gibberella, and Fusarium. Fusarium and Aspergillus are the two main ones that we worry about, because those are the ones that generally produce some of these real critical mycotoxins like aflatoxin, vomitoxin, and zearalenone. Aflatoxins produced primarily during the growing conditions. It's not a storage mold. One of the main storage molds that occurs in storage is the one that produces zearalenone. So let me just give you an example. If I harvest corn at some location and the corn is contaminated with Aspergillus or Fusarium, and maybe there's a low level of aflatoxin in the corn at harvest time, if I don't dry that corn down properly and I put it in a storage bin, those same molds may produce other mycotoxins like zearalenone and so on. So zearalenone, to my knowledge, is the primary mycotoxin that can be produced in storage. Now, of course, if you have wet grain and the molds are present, you can have continual production of whatever those mycotoxins are in storage. So the key risk management factor is getting that grain dried down to a low moisture content so the mold can't grow.
the DDG is dried up? Specifically DDG, if we're talking about DDG?
I'm talking about corn.
Yeah.
So for DDGS, the same rule would apply. If you have mold contamination and the mold is the right species, and if the moisture content is high to facilitate mold growth, you could have additional mycotoxin production. Chances of that happening are very small. Why? What's the moisture content? Those of you that are buying DDGS from Pannonia Gold, what's the moisture content? 12? You think mold grows very well at 12% moisture? No, it really doesn't. The risk factor is when you have moisture content up around 15%, then you have a much higher risk. But generally, if it's 12%, 10% moisture, it's not much of an issue. What can be done as an insurance policy or insurance factor to prevent mold growth? Are there products available to do that?
Yeah.
What are they?
Propionic acid.
Propionic acid? Mm-hmm. Good. Fumarate. Many companies have mold inhibitors. You're aware of those? Do mold inhibitors destroy mycotoxins if they're present?
No.
No. So what's the job of the mold inhibitor? Prevent mold growth, right? Do they all work the same? Are they all effective? We don't know. Good answer. Some of them do, I think. One of my frustrations as a researcher and a scientist is that, and fortunately I've been given or have the opportunity to travel to many countries around the world and talk about DDGS and feed and various other things. And the discussion we're having this morning is no different than the discussion I'd be having in China or Japan or Vietnam or Mexico or Chile. Most of the world has issues with mycotoxins. Would you all agree? The US and I think Europe, for the most part, we have them occasionally. And so when they happen, we get nervous because the risk is really high. But going back to the point I'm trying to make, mycotoxins are such a huge issue relative to feed safety, food safety. They exist everywhere, and they often occur very frequently in many countries, but we know so little about how to control them. I have a difficult time convincing companies or manufacturers of mold inhibitors or mycotoxin binders to fund research to find out if they work or not. And in the US, to make things even more complicated, if I am a company manufacturing a product that is effective as a mycotoxin binder, in order for me to call it a mycotoxin binder, I have to go through the FDA approval process, which is very expensive. So what adds confusion in the feed industry in the US is that there are effective products, but they're not called mycotoxin binders. They're some other kind of feed additive. And so it's a little bit of learning as you go and talking to colleagues in the industry to find out, does this work? Does that work? I've got some example slides if you want to spend some time talking about that. But I agree. I mean, it's one of the areas that we should have lots of technology and lots of products and knowledge about how to deal with this. But unfortunately, at least as far as I know, it's still a fairly undeveloped set of products that leads to a lot of confusion and uncertainty for the people that want to use those. Yes?
In terms of managing risk in the home lab, is there any risk in a year where you've got a high alpha toxin risk in maize, is there any risks in the following year, say with following wheat crops from those same areas? Is there carryover, in other words, from one crop to another?
Let me repeat the question. I'm not sure everybody heard. Did you all hear that? No. Is there any risk, let's say that you have aflatoxin in this year's wheat crop or corn crop. Is there a risk next year that we're going to have aflatoxins again? Let's go back to the basics. Mycotoxins are produced by molds. Molds are present in the environment everywhere. Molds are in the soil, they can be in the air, they can be in the corn stalks or the stubble in the field. So if you have those molds present, they don't go away. If the environmental conditions are right, it's like planting seeds. Okay, so there is some risk that if the environmental conditions are right next year and I had mycotoxins this year, I might get them again because the mold spores are present in the soil and if the conditions are right, it could occur. Does that mean it's absolutely going to happen? No. So there is some risk. How do we manage that? I don't know. So, other questions? Yes?
Where did the level 20 parts for baking come? And secondly, has anybody done any work to try the material was 25 parts per million? What's the benefit?
Good question. Let me repeat that again. Where did the 20 ppb standard come from is the first part. And secondly, what happens if you feed 25 or 30 ppb? The 20 ppb standard has come, aflatoxin of all mycotoxins is the most widely researched for its toxic effects in humans primarily and animals as well. So the 20 ppb is really a synthesis of all these different research studies that have led to the conclusion by regulatory scientists that's sort of the threshold of danger, so to speak. You're right, I mean, it's somewhat arbitrary. The higher the concentration, the more negative those effects are. Partly to answer your question, let me skip ahead here a bit. These are some photos, we can go back to those if you'd like, of different molds that produce different mycotoxins. These are the Food and Drug Administration in the US. These are the specifications of danger levels for different species. So to go back to your original question, where did the 20 ppb come from? It's really from a human food safety perspective that level was set. And it's based on concentrations being fed to rats as models for humans and measuring what the changes are in terms of toxic substances and its effects on cancer and so on. But what's interesting, to go back to the second part of your question, danger levels of aflatoxin or most mycotoxins vary depending on the species of animal that we're feeding. In general, regardless of mycotoxins, young animals are more sensitive than old animals. Poultry and pigs are more sensitive than cattle. And so one of the strategies that people in the US use at times is that if you have mycotoxin contaminated grain, try to sell it to your cattle feeder next door, your neighbor, because those cattle can use high concentrations much more effectively without negative effects than if I'm a pig farmer or a broiler producer. And so these are some guidelines that people in the US feed industry follow, and I don't know how similar or different they might be from Europe. They're probably very different? Okay. But it gives you at least an international perspective of how the US looks at things. Some of you that are experts in mycotoxins or deal with this all the time, teach me a little bit about what the European standards are, or maybe what some of these levels might be. Can some of you remember? Or do you choose to volunteer?
Once it's 20 parts per billion, full stop, just finished.
For dairy, it's 5?
Yeah, I think. Pigs have about 4, I think.
I'm sorry? Somebody had another comment. So 5 for dairy. When you think about many of these toxic compounds, and we're talking about mycotoxins, it's not a black and white. So if I feed up to this level, I have no problems, and if I feed over this level, I have major problems. Most of these responses are somewhat linear. So scientists are no different than people in business. It's about risk management. And depending on how risk averse a person is, that determines oftentimes what the standard's going to be. So if there's 0 tolerance for any negative effects, the standard would be 0. Is that realistic? Probably not. So in Europe, those people, the authorities that make those decisions, decided that 20 ppb for dairy is too high. We don't like that spec. We're going to be even more conservative and minimize our risk even more by lowering the standard to 5. Makes it more difficult if there's a serious outbreak or prevalence of aflatoxin, because it's harder to meet that standard, but the risk is much less relative to human food safety.
Are you aware that this is being discussed, this cutoff level, at global level or not?
The question was, is this cutoff or this standard for aflatoxin being discussed at the global level? To some extent, yes. The WHO has been having some of these discussions. Whether or not it's going to lead to a global standard, I don't know. It's one of the things that I've argued when I give talks about food or feed safety. Instead of having individual continents and countries or groups of countries having one standard and another one having another, we're in such a global society today with food products going all around the world, consumers all around the world eating sausages from Hungary, for example, in the US, or people in Asia eating foods from the EU and so on and so forth, that it makes sense to me, maybe I'm missing something, that we have a global standard. But I think the problem is different scientists, different authorities, and different governments have different tolerances for risk. And it gets into, well, if I set my standard too strict, that's going to negatively affect many people that are dealing with contaminated grain, for example. So part of the risk management strategy for a government, for example, is about how is that going to affect trade or value on top of the human food safety thing. Some other questions? Yes?
We've concentrated, and rightly so far, on the effects on threat to human health. Is there any much work done to suggest the effect of alpha toxin levels or mycotoxin levels on species performance? In other words, there's a health aspect we're discussing, but
Yes.
is there a lower level that affects performance? Is there any work to suggest at what level does the, say, performance of live weight gain, for instance, in species be affected by alpha toxins?
It's a good question. Yes, there have been many studies done in different species for specific mycotoxins. Some of you have probably read some of those studies, and here's just a helpful hint or a word of caution as you read those studies and interpret the results. Again, we do what we know, and we measure what we know or we think we know. Most of our testing focuses on, let's use aflatoxin since we're talking about. You're measuring 4 different forms of aflatoxin, B1, B2, G1, and G2. But some tests only focus on, let's say, B1. So it's the combined effect that we're most interested in, but sometimes our tests are only measuring one of those 4 pieces. That's one example. And a second example, and I've seen this in many situations on pig farms in various countries, as well as chicken farms, broilers or layers, it doesn't matter. And that is, I can read a feed ingredient or a total feed mycotoxin report from a laboratory. The laboratory does a great job with approved HPLC analytical methods, which is what we recommend. And they give the report and it says, our detection level is less than. You see the less than signs on many of these reports. So I could have aflatoxin, the 4 different subtypes, fumonisin, the couple different subtypes, vomitoxin, et cetera. And they can all have less than something. So most people would conclude, my feed is safe because I'm below these levels. Is that a safe assumption? No. What we don't understand, and I hope I'm answering your question, is that you can have very low levels or non-detectable levels, but remember that original slide I said mycotoxin experts think there may be 30,000 mycotoxins, many of those we haven't identified. So we don't know what those are, so those are unknowns. That's a risk factor. But even the ones that we do know, even if we have below our machine detection levels, which are man-made machines, doesn't guarantee that animal performance is not going to be affected. So many times we can have low levels of many different mycotoxins, and they interact generally in a negative way that can reduce animal performance. So we have to be a little careful in just not focusing on one class of mycotoxins, but we need to look at them collectively. So there are synergistic effects among mycotoxins, and I think sometimes that helps explain these unexplained reductions or slight depressions in performance that we see, even though we may go back and look at our mycotoxins and say, well, the feed should be fine because it's all below detectable levels, but sometimes it's not. So yes, there are a number of studies. Generally, let me just go through those to help bring a little bit more context to this. I'll back up here a moment. So when you feed aflatoxin contaminated feed to animals, what happens? Appetite or feed intake is one of the first things that we see that's reduced. And of course, if they don't eat as much feed, they grow slower. We can measure that on farms, regardless of species. The other thing that it does is it suppresses the immune system. So if you're in if you have animals, I'll use pigs as an example, that are in heat stress conditions, which occur in many countries around the world, especially in the summertime, and they're in a crowded environment, and you have other stress factors on them, and now you introduce another stress factor like aflatoxin contaminated feed, sometimes that's enough to trigger a health problem, a disease problem. Because aflatoxins suppress the immune system, some people, some veterinarians that I know also question if I'm feeding aflatoxin contaminated feed and I give a vaccination to protect an animal against a disease, does that vaccine work as effectively in that situation as if I didn't have aflatoxin contaminated feed? We don't know, but some people suspect that it may be reducing the efficacy of using vaccines in those situations. And of course, if you're talking about dairy cows, a reduction in milk production, part of it's a function of eating less, but it does have a negative effect on that. And it's not just dairy cows, it could be a lactating sow, it could be a beef cow out on the pasture nursing a calf, sheep, goats, whatever. And at really high levels, which God forbid we ever get to, over 1,000 ppb, it'll kill animals. Again, different species, different ages have different susceptibility. So these are the effects that we see with aflatoxin. Vomitoxin, DON, deoxynivalenol are all the same terms that we use. You're familiar with vomitoxin, right? The way I tell my students to think about or remember vomitoxin, it's a feed refusal mycotoxin. So as the concentrations increase in a feed ingredient or a complete feed of DON, generally we see a linear reduction in feed intake. Of course, if the animal doesn't eat, they don't grow as fast. Feed efficiency gets worse. In the case of pigs, which are very sensitive, more so, again, compared to cattle, and even more so compared to poultry, in real high levels, it causes vomiting. So that's where the term vomitoxin came from, because it causes vomiting. Generally, it's produced by Fusarium mold. It's a white or reddish fungus on corn. Cool, damp weather. Was it a little bit last year and a lot the year before in the US where we had a lot of cool, damp weather? We had widespread vomitoxin contamination in the corn crop, and that was a real problem. This year, it doesn't look like it's an issue. So vomitoxin, just remember that it's going to have a negative impact on feed intake, and that's about it, as far as we know. For the FDA in the US, and again, these standards I'm sure are different here in the EU. I apologize I didn't spend time doing homework to learn those before I came, but these are the guidelines our feed industry in the US follow. So I mentioned that pigs are particularly sensitive to DON, more so than chickens, and that's as a result of the threshold levels that we try to stay under to avoid significant adverse effects on feed intake or performance. Cattle, again, can handle it quite well. Zearalenone. Dan had the question earlier about storage mycotoxins or molds. Zearalenone is one that's generally produced in a storage environment if the conditions are right. The way to remember zearalenone is that it has negative effects on reproduction. It behaves a lot like the hormone, the female hormone estrogen. And one of the reasons why people worry about feeding sometimes corn, sometimes DDGS, to a pig breeding herd, because if there's significant concentrations of zearalenone, or if zearalenone's present, it'll cause abortions, it will cause irregular estrus cycles, infertility, low litter size in pigs, all the bad things that you can imagine happen in terms of reproduction. It also causes rectal prolapses in growing pigs and so on. And so this particular mycotoxin is not that much of a concern for growing animals, but it is for reproducing animals, so we worry about that. The FDA for this mycotoxin doesn't really have any guidelines. For pigs being most sensitive, less than 3 ppm, milligrams per kilogram. For finishing, breeding herd less than 2, and young pigs less than 1. Tell me about fumonisins. What do we know about fumonisins? Have you heard of that mycotoxin? Show me some hands of those of you that have heard about fumonisins. Couple, 2, 3, 4. We don't know very much about this mycotoxin. We know that it does have a negative effect on the immune system. It causes edema around the lungs, accumulation of fluid around the lungs, reduced brain, liver, and lung function. We know that the species of animals that are most susceptible to fumonisin contamination are horses and rabbits. We don't know so much about other species, and so one of the questions, and I'm probably getting off track here a little bit, was what do we know about the effects of some of these mycotoxins? Fumonisin is a relatively newly discovered one. The reason I'm bringing this up is that the U.S. corn crop, some groups have been monitoring the presence of fumonisin over the last 5 or 6 years. And it's becoming more prevalent, more common. We're seeing it in the corn crop. It's produced by Fusarium. It's a different type of mold compared to some of these others. The bad thing is it behaves a lot like aflatoxin. It's carcinogenic, causes a weakening of the immune system and so on. There are 3 types, B1, B2, and B3. Most of what we measure is B1, but this is one to be aware of, and it may help us explain from time to time some negative effects on performance, even though maybe our aflatoxin or vomitoxin or other mycotoxins are normal. Because we don't know very much about its effects in various species, these action levels or threshold levels of concern are extrapolated from the most sensitive species, horses and rabbits. And we think that poultry are the most tolerant, followed by cattle. Pigs are kind of in the middle, and again, horses and rabbits are the most sensitive. So, time for another little quiz, and then I think we'll take a break for 15 minutes. The quiz question is, if I have aflatoxin contaminated feed that I'm going to feed to animals, what mycotoxin binder can I use that's been shown to be effective in detoxifying or binding aflatoxin specifically? Do any of you know? The answer is many of the clay-based products are very effective. Have some of you heard about a compound called sodium bentonite? Where do we use sodium bentonite in a feed mill? What's its purpose primarily? It's a pellet binder. It's fairly inexpensive. I don't know how much it's used in Europe, but it's very commonly used in the US. So sodium bentonite is not that expensive, and it's very effective as an aflatoxin binder. The mistake some people make, though, is they assume that just because it binds aflatoxin, it works for all mycotoxins. That's not the case. So fortunately, the good news is that for aflatoxins, we have some very effective binders that are not that expensive, and they're the clay-based materials. They come under a variety of trade names sold by various companies. And so you're going to be looking for active ingredients like sodium bentonite or things that are clay-based. And they'll bind, depending on the product, somewhere between usually 75 to 100% of the aflatoxins if they're present. And that's a good thing because then the animal doesn't get affected by this contaminated grain. So that's a management strategy if we know that we have those problems. What about, is there an effective mycotoxin binder for DON or vomitoxin? Does anybody know? I'm sorry? These other mycotoxins are more difficult. I can tell you, and I can show you some data of some US products, maybe some of these are also available in the EU as well. But for DON or vomitoxin, the yeast-based products seem to be fairly effective. Yeast cell walls or yeast-based products seem to have some effect, but if the DON or vomitoxin concentration gets too high, generally more than maybe some cases 3, certainly at 5 ppm, they're not effective at all. But at lower levels, they seem to provide some binding or some benefit, and it varies depending on the product. I don't know of anyone that's particularly effective for zearalenone. And fumonisin, as I mentioned, is a bit of a question mark as well.